Then cell organelles is different type of the cell organelles. That is mitochondria, plasmids. These are the general type of the cell organelles. So another kind of the variations, that is a different type of the cell organelles. That means endoplasmic reticulum, Golgi complexes, ribosomes, lysosomes, peroxisomes. These are also different type of the cell organelles. So first of all, we discuss about the ER, that means endoplasmic cells. Endoplasmic resistance cells. So ER. Then that ER cells, that means these are, first of all, these cells is discovered by the Porter endoplasmic reticulum cells is discovered by the endoplasmic reticulum ER cells is discovered by the Porter. So in this different type of the endoplasmic reticulum cells, that cells is completely tube-like structures. For example, different type of the structures, they have the different type of cells. In these type of cells, linked or connect with the 1 to another one. Connect with 1 to another one cells. The cells is interconnected one cell to another cell. So, these are the general type of the cells. That cells is different type of the cells, then the different variations of the cells. This cells is called the different type of the cells is called the, then one type of the cells and different type of the cells. These cells is discovered by the Porter. When this one is different, then that it has in two layers type of the cells. That is the, when this one is the depending of the layer, that layer it is called the lumen. Then this is the lumen type of the layer. Then another one, that is the, then this what it is called the nucleus layer. Then in this of the cell membranes. Then that is the cell membrane. So in this part, in this type of the part, it is the endoplasmic reticulum. In this type of the endoplasmic reticulum is connected with the one to another cells of the space of the cells. That is the different thing of the cells. So in this, in different type of these cells is going to the different type of the structures. Then different type of structures. These are the just like tubes-like structures. These cells is like in tube-like structures. These tubes are connected with one to another ones. So these tubes flat-ended cells, that the flat-ended source or flat-ended vessels type of the cells. In these type of the cells, that is constructed with the different type, different to one to another one. There, these are the generally L-8 to halo type of the structures. Different, that means weak halo type of the Then these leaf halo type of structures, the nucleus is completely then convert or connect with the envelope type of the structures. In these type of the structures, envelope type of the structure, one to another side. Then different two sides of the simple way of the structures. In these of one type or one side is the cell membrane and another side is nucleus membrane or nucleus. So here the tube-like structure one, the yaw of the interspace, that is called the lumen. So in these are the enclosed, the complete enclosed of the cell membrane and the nucleus stages. So here E are completely then resistance. They can region of, they can carry into the different type of the E or lamellas. Then these E or lamellas, that is the two types, that means rough E or at the same time smooth E or. Same. Then going to the rough E or, that means it has some different type of the rough structure of E ors. Then the tube-like structure, they have the formation roughly structures, that is the rough E ors. Rough E or structures. Then the cells, developed cells. These rough E or structure cells is developed cells. Rough E or structure is very developed cells. Then these are the engaged in different types of the proteins, or it is in completely very developed cells, and it is the rough E ors also involved in synthesis of the proteins. Synthesis of the proteins. So here different type of the layers, that is the ribosomes. It has in the attachment of the different types of the ribosomes, the different type of. Then the ribosomes is engaged completely, they are involved or synthesized or ATS ribosomes. Then ATS ribosomes also. Then the attachment of the surfaces in the cell membrane layers. So here it is also then different way in the smooth ER. That the smooth ER, it is completely consistent with the rich amount of the glycogen. It is in the synthesis of the protein synthesis. It is the glycogen. It is in glycogen. So then it is formation of then the vessels. That is the proteins or lipid type of the layers. Then the proteins and lipids. They have the different type of the proteins and the lipids. So here it is then the make. It has the made up of the different type of the fats. That is the different type of the fats type of the cells. Then it is also three variations of the cells. The three variations, it means that the smooth ER, it has in three types. That is the different type of the three types. One is the tubeless line of the structure, that is the called the cisterns. Then tubes or bags. Then tubes. So cisterns, that means a different type of the structures. That is the cisterns like of the structures. These are the general flattened like of the structures, and these are also long or small cisterns. So then the bags, these are the general like structures, that is the small or long type of the round surface, it is called the bags. Then the tubes, this is undifferentiated, that means different type of the shapes or different type of the structures. The tubes is different type of the structures. So these tubes is also different in shape or different type of structures. So where the endoplasmic reticulum is completed in the mechanism, different type of the enclosures of the function. So it is the function of the endoplasmic reticulum. Endoplasmic reticulum, functions of endoplasmic reticulum. It has in the completely different, that means synthesis of the proteins and the functions. That functions is the synthesis of proteins. Synthesis of proteins. At the same time, exchange and translocations of different types. It is in exchange. That exchange, but that means variations of the different types of the substances exchange. Different type of substances exchange. Then different type of the protein synthesis, that means rough ER protein synthesis. The attach of the ribosomes. Rough ER protein synthesis, attachment of the ribosomes. It's carrying different type of the secretomic or protein synthesis. When the synthesis is the proteins, the synthesis of secretory proteins also it can be synthesized. Then also again it is the complete translocation or exchanges, where exchange different variations of the different substances. The substances going to the one tube to another tube or one place to another place. That is one of the exchange of the substances. Then the different variations of the smooth or rough, these are the complete quite opposite manner in the directions. That is the development of one of the proteins, one of the nitrogens. So then these are complete different things. That is the lipoproteins or glycogens also synthesized. In this is the function is also another function is glycoproteins. Glycoproteins and different type of lipoproteins also. Lipoproteins also synthesized. Then the smooth or rough, completely they can go into the different type of the lipid collaborations with the different type of the lipid variations. Then the second one is ribosomes. The second one is ribosomes. Ribosomes. Then the ribosomes, it is discovered by the Pallada. Discovered by the Pallada. These are the complete cells. These are the complete, then the small cells. Then the different type of the small type of the cells. Then the compared to the cell to cell, these are the very small cells. The ribosomes is very small cells, small ribosomal cells. It is complete in the universal cells. The ribosome cells is universal cells. These universal cells completely in the primitive type of the cells. The ribosomal cells is universal cells. At the same time, these cells is completely primitive cells. Primitive cells. So these are present in complete in all types of the cells. That means eukaryotic type of cells, eukaryotic cells, at the same time prokaryotic cells. Eukaryotic cells as well as prokaryotic type of the cells. These are the special type of in the shape or structures. That is the different type of the spherical like structures. In the structure is in the ribosomes is spherical shape. Spherical in shape. So here are different type of the models. That means different type of models. That means it has in the made up of by the ribosomes is made up of by the RNA and proteins. RNA plus proteins. Then the RNA and proteins. These are completely then each ribosomes they complete the involve the different type of the participations in the substances. Then the each kind of the ribosomes they have the two types of the ribosomes things. Then the two type of ribosomes that means 80S ribosomes at the same time 70S ribosomes. So then ribosome is also one of the different type of the structures that of the head-like structure. Then in the different type of the ribosome structures. Then different variations of the structures. Then it is called the head like type of the then structures. So then these type of 70S ribosomes at the same time 80S ribosomes. These are most involved of the different variations. Then going to the different type of the 70S and as well as 80S ribosomes. So here 80S ribosomes most of the complete that involves in the eukaryotic cells. Then 80S ribosomes it is present in the eukaryotic type of the cells. Then 70S ribosomes, these are the prokaryotes. The 70S ribosomes is present in prokaryotes. So in these 80S, 70S ribosomes, it is also different type of the substances or sub-units also. It has in different type of the sub-units. That sub-unit it is called to the 70S ribosomes. It is 80S ribosomes. It is two varieties of the sub-units. The sub-units, 80S ribosomes, it makes into the 60S plus 30S ribosomes. So here it is the 60S is very longer in 30S ribosomes. So then again the 70S type of the ribosomes, that is the it is also in the 50S ribosome plus 30S ribosomes. 50S ribosomes is longer than the 30S type of the ribosomes. So here then it has completely then the way of or use of then the that means information. That means information that genetical information. It used completely then the ribosome. That means RNA ribosomes. It goes to the different type of genetical information. It's used to the genetical information. It used to the genetical information. So then in these type of the genetical information, it has in the mRNA synthesizer. mRNA synthesizer. So it is the participations of the, that means chain, polypeptide chain. It is the involved in the polypeptide chain. involved in the polypeptide chain. So then it's completely the different type of the genetical variations of the ribosomes. Any different of the genetical variations of the ribosomes. These are the general functions. It is a provide of different type of the enzymes. It is provide enzymes. It's provide enzymes. They can also synthesize protein synthesize. It is also used in the production or provide of the enzymes. At the same time, protein, it is also used in the protein synthesis. So these are the general about of the ribosomes. The ribosomes discovered by the Ballada. These are the universal cells and most primitive cells. These cells is a different type of the cells in among the all cells. That is the eukaryotic type of it. The ribosomes is present in eukaryotic cells as well as prokaryotic cells. So here these ribosomes is spherical like structures of the spherical like structure in the shape. Then these are connect with or constrict with the RNA and proteins. So these different type of the RNA, this RNA is going to the genetical information. It gives to the genetical information. That genetical information is provided to the different type of the in RNA synthesized genetical material. Then these type of general RNA proteins is going to the two ways. That is the 90S type of the ribosomes at the same time 70S types of the ribosomes. These two variations also two different type of the cells. That is the eukaryotic cells. 80S ribosomes is present in the eukaryotic cells. 70S ribosomes is present in the prokaryotic cells. So then these different type of the cells, that is the 80S ribosomes is subdivided or subcells. It is in the 60S plus 30S type. Then 70S is also 50S plus 30S. So these are the different type of the cells. Then other topic is that is the Golgi complex. Other one is Golgi complexes. Golgi complexes. It is discovered by the Cavello Golgi. Golgi is discovered by the Cavello Golgi. So it is also different type of the structures. The different type of structures, that means then these are called the secretory vessels. These are called the secretory vessels. Then these interlinkation with the different structures. It has a tubes-like structure, flat-ended to tubes-like structures. Then different type of the structures. That is the different type of structure is cystin, and at the same time lumen. The end of the cystin is called to the lumen. Then it has to the different types of bags-like structures. These are called the bags. Then these called the completely saccus. These are the saccus. That is a complete different type of the system. That means Camellia Golgi is whenever the discovered in a different type of structure Golgi complexes. This is the complex method. That is the different methods is in only one way or in only one kind of the manner. So here different type of the structures. So these are the complete, it is also Golgi complexes is known as idiosomes or dictyosomes. It is called to the Golgi complexes is also called to the different type of the names. That is the idiosomes. At the same time, dictyosomes. The idiosomes, dictyosomes, these are also different type of the manner. Then the dictyosomes, it has in the cystae at the same time sacules. Dictyosomes it has in the cystae and that means saccule. Then this like a structure and sacules. Then it is in complete in all plants. The dictyosomes is present in all type of the plants. Then the remaining dictyosomes is also different variations. That means the cystae, vascules or also then bags. These are the different type of the then these cystae, that means these are surface one to another one parallel like systems. That is the piles or black into the different building. It is in the buildings. Then the bags is completely the small or spherical round like structures. Round spherical structures. These consist of the cellulose, hemicellulose. It has the cellulose and hemicellulose type of the structures. Then the sacs completely it is in the different ways. So cellulose, hemicellulose, and sometimes it has in the proteins also present. So then these are the different way of the Golgi complexes. Different type of the structures involved in the one kind of the way. Then going to the functions, the Golgi complexes functions. Different type of the functions. These are the synthesized the cellulose and hemicellulose. Synthesized cellulose synthesized by the cellulose and hemicellulose. At the same time, production of all membrane. It is in the production of all membrane. That means different type of the membranes. It is the most important role. It is the metabolism. The Golgi complexes is most important in the metabolism. These are the formation of also. That is the important one is the cell division. That is the cell division. The metabolism as well as cell division is the most important functions in the Golgi complexes. So then these are the different type of in the all cystae, sacs. These are each and every one is different to one to another one. So in these different things they can use in the only one kind of the manner. The different type of the cells is in only one type of the manner. So the Golgi complexes, it is the complete the structure of different things or different type of the cells is going to the one kind of manner, that is the complex. So these are the general complexes. These are completely different variation of the lipo or lipid type of the metabolism. So then this cells is then the different to the complete then the connected with the celluloses and most of them the hemicellulosic type of the cells. So here each and every one is also they connect with the another one. At the same time they all are also in the manner different type of the things. The bags is also one of the consist of different type of the things. At the same time sacules also different type of the things. They have to the mention or they have into the structure, that is the chemical structure, that is the involvement, that compound is synthesized as well as the compound is made up of is also different things. So that's why in this complex manner is in the Golgi complexes. Okay.